let's get started. Hi, everyone, and welcome to the first session of our High-Affinity Talks. We're excited to launch this new series where we spotlight leading researchers in the biologic space and invite them to share their latest insights, breakthroughs, and perspectives on topics shaping the biologics field today. Today, we have Mette Soendergaard, who is the CSO and co-founder of Cell Origins, a boutique CRO specializing in peptide and antibody discovery through phage display. And she's also a professor at Western Illinois University, specializing in peptide discovery. Today, she'll be presenting her topic on going beyond binding affinity and talking about what really matters, transitioning from phage discovery into in vivo and in dynamic molecular states. After the presentation, she will also be joined by Sam Pozlowski, CEO of My Laboratories, a company that focuses on biologics discovery software, to dive deeper into that topic. So just some housekeeping here. This webinar will be recorded and sent out automatically afterwards. And then we'll also have time for questions at the end, but feel free to submit them as we go on. Okay, great. So I'm very pleased to welcome Mette for her topic.
Thank you very much, Monica, and thank you to both of you and your whole team for inviting me to give this webinar. I'm going to talk about going beyond binding affinity today when it comes to peptide discovery, but I just want to preface with saying that it's not that I don't think that binding affinity is important. I just think that it's equally important to look beyond just binding affinity and look for how we can optimize peptides for other kind of what I call behaviors, basically. So I just want to start with a little bit of background, I don't know if everyone is familiar with phage display who's joined us. So phage display is a technology that's used to discover peptides, also antibodies. So everything that I will be talking about today from my own research and the research at Cell Origins is about peptides, but we could be doing the same things for antibodies. So antibody fragments like scFvs, Fabs, nanobodies, and even other proteins like antibodies. So the technology of phage display was developed by Dr. George Smith at the University of Missouri back in the 80s, so the mid-80s, and he was awarded the Nobel Prize in 2018 in chemistry for that development. So what the technology is based on is that we take filamentous phage, which are viruses that can infect E. coli, and genetically modify them to display a foreign peptide on one of the coat proteins. These phage particles are then assembled into libraries that typically have somewhere between 10 to the 9, maybe up to 10 to the 11 different types of foreign peptides. And we can then use a library like that to screen for a certain peptide binding to maybe a protein biomarker or any molecule, really. So how this is done is by doing what's called biopanning or affinity selection. You'll hear me probably use both terms throughout my talk. But biopanning is really just challenging the phage to see which ones bind to whatever the target is. And again, I would say most of the time when we're talking a target, it's going to be a protein, typically a cell surface protein like a receptor, but it could really be any type of molecule. There's been phage display selections developing peptides and antibodies against metal ions, other inorganic materials, as well as carbohydrates and even lipids. So in theory, this can be done against any type of molecule. So the phage display library is incubated with the target here, which I typically call the biomarker since I'm working in oncology. So we would typically want to find a peptide that binds to a protein that's associated with cancer somehow. We wash away all the non-binders, and then we elute the binders. We amplify those in E. coli and then subject that collection to another typically 3 rounds. So a total of 4 rounds against the actual protein target here. After the last round, we then analyze the phage to identify the peptides. We do that with the NGS, and we also do that with more advanced bioinformatic analysis. One of the things that we find very helpful is taking the sequences and, of course, sorting them according to frequency with the kind of notion that the higher or the more often a peptide occurs, it's likely that it's been enriched in this biopanning protocol. However, it's really not the only thing that we want to look at. What we find equally, if maybe not more beneficial, is to cluster peptides. So we identify amino acid motifs and basically go in and looking for pharmacophores, which should be indicative of having peptides or peptide families that bind to the target. Okay, so I also want to give a little bit of a history lesson today on peptide phase display. I recently went to the Peptalk conference and I thought it was really fascinating to see where the field is now, but also how far we have come. So when we started back in 1985, when George Smith developed phage display, and since then, a lot of different phage display libraries have been developed. It started out with the linear peptide libraries, and nowadays the cyclic libraries are really, really popular, basically for their stability, but also very much because they tend to deliver higher binding affinity. So it might be a surprise to some that the first cyclic library was developed already back in 1992. But I also want to highlight a really important landmark study here by Heinz et al. In 2009 that developed the first bicyclic libraries. So these are in the clinic now, in clinical trials, and have really been groundbreaking in looking at how peptides, they bind with protein interactions. So that kind of overall structure and many different derivatives of that have become more and more popular. So I also, I'm going to talk a lot about biopanning today. So that's basically half of my presentation is going to go into different biopanning strategies. And one of the things that we really focus on is doing in vivo or ex vivo or cell biopanning. And I don't want anyone to think that this is, we think this is new, because it's not, because it was already basically kind of the groundbreaking work was done back in the mid-1990s by Pasqualini and Ruslati. In 2002, Arab et al. Did human biopanning in patients. So this has been done before, but it's really looking at how we can achieve the best peptide binders and pharmacokinetics and so on using these different strategies. So another thing I don't want to leave out is next generation sequencing employed to phage display. So this is, of course, really relevant to the talk today and to my laboratories. So before next generation sequencing was used for phage display, I might be dating myself here. We're looking kind of like I was doing my PhD down here in the like 2009-ish era, like 2014. And back then, most laboratories were still collecting colonies, E. coli colonies, and isolating the phage, identifying it with Sanger sequencing, right? So as a graduate student, you would pick hundreds of colonies, send them all for Sanger sequencing, and there was a lot of work. So with next generation sequencing and be able to go really deep with the data, that has allowed us to get enormous amounts of data, right? It also has complicated things in how we analyze it, of course, but it's really given us an enormous amount of data in addition to kind of save the graduate students in the lab a little bit. And we don't want to ignore machine learning either. There are many different platforms out there right now. Some are developing peptide phage display AI. Some have already done that. There's many different companies looking at different aspects at this. Overall, trying to add to the pipeline of how we select peptides. How do we identify the best binders before we move further and start screening our hits? Okay, so even though we've come really far compared to where we started in 1985, there are a lot of persistent challenges. And if we look at kind of the overall strategy or steps for discovery here, we can see we, of course, start with a target hypothesis. Most scientists want to know, are going to choose like a protein. This protein might be involved in cancer, it's overexpressed in this type of cancer. This typically starts out at universities, right? Basic research can then decide to do display selection. So I'm going to talk about phage display, but I'm not ignoring that, of course, there are other ways of doing this as well. Screening of hits, lead optimization, and then we get into animal studies. And where we see things failing, that's at this step. So once a peptide gets challenged, basically, in this really complex biological environment, now we start to see that the peptide is no longer performing as we wanted it to. So we can we can have too rapid clearance. If we're developing peptide drugs, we want them to get to the cells, to the tumor, for example, and of course have enough time to elicit an effect. Very often we get off-target binding to isoforms or paralogs from different proteins. And then what's also very common is now no longer the peptide binds. So we lose binding. And what I want to show with the talk today is that we believe one of the reasons why we lose binding is because biology is complex, right? We tend to think of a peptide binding a single molecule, and that's also what happens once we go in vivo. But in reality, here we have protein complexes, glycosylation, and a lot of other things going on that I'm going to touch on later. So as we're setting up a phage display experiment, basically a biopanning strategy, right? We want to enrich for certain peptide properties. So this is where we at Cell Origins and also in our previous academic work have spent a lot of time optimizing biopanning strategies to make sure that the properties that we're actually enriching for are the ones that we're getting, right? So the classic way of doing phage display is on a plate against a typically like a recombinant target, right? A recombinant protein that we can buy online or produce ourselves in the lab. And when we're doing this, we can very easily get great peptides with great binding affinity, very low KD values. But we're also really enriching for the peptide properties in the way that we're getting a peptide that binds really well to that recombinant protein on that plate, right? It does not often translate into animal studies or in vivo work. So in the ideal world, right, we would all be doing human in vivo biopanning like Arab. It all did back in 2002. I think for most of us, that's kind of out of reach for obvious reasons and also funding, of course. So that leaves us with what we have kind of in the middle here, right? So we can do cell studies. That could be cell lines, but it could also be cells kind of harvested from tissues and grown in the lab, ex vivo studies, and then, of course, so also in vivo. And what our background is in oncology. So typically we want to target a tumor, maybe a specific biomarker in a tumor. So here we're looking for a mouse tumor model and basically challenging the phage display library to access the tumor in the live animal. So as we're doing this, kind of setting up the biopanning strategies and thinking about how we want to go about this, I think it's really important to distinguish the molecular binding that we see on a plate, right? We immobilize a recombinant protein. We go for binding. We might do SPR to get some KD values. And it's really, what we're looking at is how that peptide binds on that plate, right? It does not reflect how it will perform in vivo. I already touched upon this, but what I really want to highlight is the accessibility of the target. Most proteins exist in complexes with other proteins at the cell surface, right? We also have glycosylation patterns. We can have different types of lipids in the cell membrane forming lipid wraps. We can have overexpression, underexpression. So it can be really difficult to recapitulate the environment of a mouse model or an animal model on a plate. In addition to that, we can have fast clearance or degradation of the peptides. So an example I just brought along, just to kind of emphasize my point here, is carbonic anhydrase, so 9. And we can see here CA9 we know that it's overexpressed in certain cancer tissues. We also know that it has low expression in some normal tissues like the stomach and the small intestine. And when we look at carbonic anhydrase 9, it sits in the cell membrane and it sits in the complex, like most cell surface proteins do. And I think it's fairly straightforward to see here that if we are biopanning against the isolated form of CA9, we could end up with peptides that bind to the interaction interface here over to its binding partner CD147, right? Now we're going to lose binding in vivo. Additionally, CA9 has this large proteoglycan domain that's very flexible, but it's also glycosylated. Of course, we can mimic that in recombinant proteins depending on the expression, but generally this can be harder to replicate compared to the in vivo state. On top of that, many cancers have different glycosylation patterns than normal cells do, even for the same protein. So these things all complicate matters, and basically, I think it emphasizes why we really need to have our protein target in the most native state that we can when we undergo or when we carry out the biopanning. Okay, so just real quick, I wanted to do like a little case study basically, where we took a peptide from phage display and then all the way to imaging, which was the goal. So this is older study. This is in fact for my PhD. And the goal here was that we wanted to identify a peptide that could bind and target ovarian tumors and image them using radio imaging. So we're doing a lot of radio labeling and SPECT and PET imaging and things like that. So here, the strategy was to first pre-clear the phage display library in mice. Actually, I should take a little step back. I'm sorry. I want to explain why it says blinded in vivo phage display here at the top. So the classic or traditional way of doing phage display is selecting a target, a protein of choice, and then going through the biopanning strategies. There's nothing wrong with that, but we wanted to see what if we don't try to target a specific protein, but we are selecting for a peptide's optimal function. We wanted to bind to these ovarian cancer cells, or human SCOF3 cells, the cell line, and we wanted to be able to image that. So we didn't know what the target was. We just knew we wanted to bind specifically to those types of ovarian cancer cells. So we started out by pre-clearing our phage display library in normal mice, and then we went on to what we would call positive selection. So 4 rounds of that in tumor-bearing mice, excising the tumors, and then creating single-cell suspensions. At this point, we also wanted to make sure that we weren't kind of by mistake targeting some of the other types of cells that we typically see in a tumor. So tumors is not just cancer cells, right? We'll have stromal cells, we're going to have immune cells, vasculature, and so on. So we isolated the cancer cells using antibodies against known ovarian cancer biomarkers, eluted the phage using a detergent, amplified those, did DNA sequencing. This was with the old-fashioned way of picking colonies, identified the peptides, and then finally took those and screened them against the SCOF3 target cell line and also a normal, basically negative control cell line. So from all of this in vivo selection, we ended up with a peptide we called a peptide J18. That's from my co-founder, Jessica Newton-Northup. She started the experiment, so I named it J after Jessica. So the first thing that we do, and I think many other scientists do as well when we identify peptide hits, is to go in and identify or figure out the binding affinity. And again, I'm not talking against binding affinity here. I think it's very important to go in and figure out what is the KD value, or in this case, we're looking at an EC50 value because we're looking at a cell line. We still don't know what the target is, right? We're measuring basically how well does this peptide bind to our target cell line. And we saw that we got an EC50 value of about 22 micromolars, which if you've ever done anything like this, you will not be impressed by that number. And we weren't really either. So I think on a relatively good day, you might call that moderate binding affinity. It is definitely not a value that will stick out to you in the lab and think you found like the best peptide, right? There's not the holy grail EC50. So we weren't completely discouraged yet, though, because we do know doing in vivo phage display always results in peptides with lower binding affinity. It's just a very strong trend that we've always seen. So as long as we're in the lower micromolar area, we're still optimistic that we can develop the peptide further. One other thing we wanted to look at was peptide stability. So for anyone who's worked with peptides, you know that they are just inherently unstable, especially in vivo. We measured the half-life here in mouse serum, and we saw it was 5.2 hours. And that is actually really great for a natural peptide with no modifications on there. So we were kind of happy to see that, because even though it might not have a high binding affinity, then at least we had high stability, which may be able to lead it to the tumor and stay there for longer. So we decided to go and challenge the peptide, like we like to say, and see like how does it actually behave in a mouse model. And what we saw here was that we actually were able to target it, right? So we labeled the peptide J18 with indium-111 for SPECT imaging, injected it into a tail vein of a mouse. And after 1 hour, we could see here that we clearly can identify the tumor, right? We get kidney excretion into the bladder, the urine in the bladder here, completely normal. All peptides do that. We cannot get rid of kidney excretion because we do need the peptide to leave the animal, hopefully eventually the patient, right? But what we were really, really happy to see is we have no off-target binding, right? We got the tumor and that's it. There's nothing binding to what looks like vasculature, stomach, intestines, liver, anything like that. So it's very clean imaging, basically, that we were very happy about the day that we saw this image. Looking over here, this is a block. So basically, we're testing for specificity. Here, we pre-injected the mouse with non-radiolabeled peptide to see if that would block the tumor uptake. And we could see that, in fact, it did do that, right? So we knew that we had specific binding. Okay, so overall, we haven't really taken this study further. I graduated with my PhD, and we kind of left it like for a lot of people, right? Kind of left it at that. But we knew that this type of doing blinded in vivo phage display can result in peptides that have great properties, right? That behave really well, because we are mostly selecting for function, basically in this case, binding or targeting, rather than for a specific protein. So for the work that we've done at Cell Origins recently, we took a similar approach. So here we wanted to go and target phenotypical changes in cell lines. So the idea here was that cells, they change their phenotypes as we're culturing them in the lab. Anyone that's worked with cell culture, cell lines, know that we cannot keep passaging them for very long. They're going to start changing. They're going to look differently. They're going to maybe detach from the cell surface. There's definitely changes somehow to the cells. And we wanted to see if can we do phage display against these cell lines and identify peptides that can differentiate between young cells and old cells, and also between two different cell lines. So the targets here were LNCAP cells, which is a prostate cancer cell line, and then also HEC293s. These were basically kind of our control cell line. The idea was getting binding or peptides to the LNCAPs and not to the HEC cells, and being able to differentiate between low passage or high passage number, basically young or old cells. So we did two different types of biopanning strategies here. We did the classic way of doing phage display, which is incubating the library with the target. We're washing away non-binders, and we're eluting binders, and then subjecting those to another round, just like I basically went over at the beginning of my talk today. Then we also did what's called a depletion selection. So here we incubate again with the target, but then we let it interact for just a little while, and then we take everything that does not bind and subject that to another round of binding. And this might sound weird, but there's been studies that have shown that we can kind of select phage or peptides that bind to targets that are in lower copy number in this way. The idea behind this being that we're washing away after a short amount of time everything that is binding to high copy number targets, right? On a cancer cell, that could be things like maybe EGFR or anything other like known overexpressed cancer protein, right? So here, when we're collecting everything that doesn't bind immediately and then subjecting that population to another round of selection, allowing them now to bind for a longer period of time, that, at least in theory, should allow us to select peptides that bind to low copy number proteins. So after we did all these selections, of course, we needed to analyze it. We did NGS, and then we did some analysis. And this was a custom program that was written by a collaborator, Dr. Austin Prater. And he got the job basically to go and figure out how can we best analyze all these different populations, all these different selections. So I have not shown all the data here because that would just take up a lot of space and a lot of time. But what we can see is that we did manage to, for example, for the LNCAP cells, we can see that we identify unique peptides that were unique to that elution method, the traditional phage display, and also to the one that we call the depletion strategy. We did that for both the low passes number and high passes number cells. And when we compared the low versus high, we also identified unique peptides. And if we're looking down here at the bottom row, we could also see that was the case for LNCAP cells versus HEC cells. So in that way, we were successful. We can see, okay, we did actually select peptides that were specific to each of these conditions. Then the next step comes because, in my opinion, phage display basically always works in the way that we always find unique peptides. But now we need to challenge them and see if we can get them to bind. Again, I have not shown all our data here for just simplicity. I've chosen to use one peptide as an example. We have an enormous amount of data, and some of it we're not even done kind of analyzing yet, okay? But basically what we did here was we wanted to see, of course, if the peptide actually bound to the LNCAP cells versus HEC cells. And we did a phage binding assay. So instead of going in and having the peptide synthesized, we had it expressed on the phage. It's quicker and it's also more cost effective. And then we go in and measure binding via qPCR because the phage particles have their DNA on the inside. So what we could see was that we did in fact get binding specific to the LNCAP cells. Here we see a control phage and our peptide, and we got specific binding to the LNCAP cells, at least compared to the HEC cells. We also saw that for this peptide down here, that we got binding that was basically lost as these cells were getting older. So we take these cells all the way out to passes number 40, which I don't recommend. If you work with cells, you know not to do that, right? But you can see here why, because we lose some kind of binding. Something happens phenotypically to the cell surface that makes the phage no longer be able to bind. So we wanted to figure out what was going on. And the way that we can do this when we do these types of blinded phage display experiments is by basically go and get the peptide bind to the cell surface. We can do a pull down assay and then mass spec and hope that we pull down our target, right? That would be kind of the most straightforward way to do it. But we really wanted to get a more holistic picture, basically, if you can call it that, of what was going on at the cell surface. Because we believe that the reason why a lot of peptides lose binding is because the cell surface is very complex, right? Biology is complex. Many other things are going on. So we wanted to see which genes are basically involved in binding or result in loss of binding. So we designed an experiment here where we use a CRISPR whole genome knockout library against human genes. And we took that and infect or transfected LNCAP cells here. So we had two populations of cells, one that was transfected with Cas9, RFP, red fluorescent protein, CRISPR genome knockout library, and then one without the knockout library. Incubated these two cell populations with fluorescently labeled phage. We had controls here and the phage that we wanted to try out that we knew where we lost binding over time. And then we did flow cytometry, sorting the cells. What we wanted to see was if we have binding when a gene is not knocked down, but we lose binding when that gene is knocked down, that must mean that that gene has something to do with the binding, right? Exactly what it would be, we didn't know yet. But that was like the idea behind this study. Do we see loss of binding as this gene is knocked down? So again, enormous amount of data to analyze. And we did NGS of that, sorted all those individual cell populations, and then organized it into the gRNA with the gene IDs. So we can go in and identify, are there gene clusters or certain genes that are responsible for loss of binding? And this point, we see, like, if we were, I don't know if we would call it lucky, but if things were straightforward, we would have seen just maybe, okay, when we knock out this gene, this cell surface receptor, now we lose binding. But it was not that simple. So this is one of the reasons why we're starting to realize really how complex things are. So we identified several gene clusters. I'm going to talk about two of them here, the two top ones, basically. And the top one here were genes that had to do with afrin, EPH receptor signaling, as well as semaphorin and plexin. These are known to be basically in cell adherence, signaling to cells that are next to each other, neighboring cells, and attaching them to each other. Also, receptor clustering, as I mentioned before, a lot of cell surface proteins exist in clusters, right? So basically in protein complexes. Cytoskeletal remodeling pathways, also indicative of the cells getting older. If you've seen old cells in the lab, you'll notice that they start detaching, right? They're not attached as well to each other, and they're not very well attached to the surface either. And they kind of change their overall morphology, so they look different. And then finally, stabilization of receptor assemblies by adapter signaling proteins. So we're looking down here kind of below the signal transduction pathways of these identified interactions up here. This kind of pointed in the direction to say, okay, could it be that one of these is our target, or is the peptide maybe binding to an interface here? We need expression of these cell surface proteins, but we need them to be in the complex kind of configuration for the peptide to find its epitope. So we look at the second cluster as well. And here we found something that's kind of surprised me, to be honest, because I wasn't expecting to see anything with lipids, and specifically not peroxisomes. I'm going to admit that this is not my area, so I had to look this up and be like, how is this related to the cell surface? So peroxisome-dependent lipid metabolism very much basically dictates or determines which membranes are in the, which lipids are in the plasma membrane. So when protein complexes assemble into lipid wraps, for example, it's very much dependent on the types of lipids. Also, plasma membrane lipid symmetry and cholesterol handling, trafficking and recycling, taking proteins from the cell surface back into the cells. And then again, very interesting, receptor clustering, right? So getting those cell surface proteins into their complexes, and then functional microdomains that is also including something like lipid wraps. So now we know if we lose a specific composition of the cell membrane, we also lose binding. So not only do we need certain proteins to be present in certain protein complexes, but we also need a specific membrane lipid composition before we can get peptide binding. So this was very surprising to us because we hadn't seen that before. And as far as I know, I haven't seen any other published articles that have described this before either. Okay, so quickly, I just want to look here at some more data. Just got a few slides left of data. So we wanted to look at the gene expression level as well. So we were looking at the cell surface receptor before, or cell surface, and looking at if we lose this protein, we don't get binding, right? So does that correlate to the gene expression at the RNA level then? So we decided to go and do RNA-seq, comparing old cells to young cells. And if we look over on the left here, these genes that had the highest and lowest log2 fold changes were not the same ones as we identified with CRISPR, at least generally speaking. We can see the CRISPR genes we actually see here at the bottom of the volcano plot, or most of them are. So at this point, we're like, well, what is really going on? And of course, looking into these genes that were changed on the left here, old versus young. And what we saw was several of these are responsible for glycosylation, so different types of oligosaccharides on the cell surface, and also the glycocalyx, which is the different carbohydrates and kind of networks that we have in the extracellular matrix. So when we take all of this together, right, we can see that the CRISPR screen identify these kind of networks or complexes We know that we need to have certain protein complexes form. We know that like binding is dependent on those adapters as well, but we also need to have a certain composition in the plasma membrane of the lipids, right? And when it came to the RNA-seq data, we see, well, in addition to that, one of the things that is changing with age is the glycosylation patterns. So glycosylation and very much the glycocalyx is involved in cell movement and, of course, attachment to the cell surface and attachment to other proteins, right? So in this way, that makes sense. It correlates with the literature that as the cells are getting older, we know they lose binding to the cells into the plate. And this could be, in part at least, because of the change in glycosylation and glycocalyx. We also saw lipid handling and membrane composition there as well. And overall, this got to have to do with adhesion and surface organization. So we're not done with this data. We're still analyzing it. I would love to, if anyone has any input. This is still very much a working hypothesis. I want to put that out there. We're continuing this research, but what it tells us so far is that we get an altered epitope, right? As the cells are changing, the epitope changes, so we no longer get binding. This is also what we very much could see once we challenge in an animal model, right? The idea that the peptide binds to a single molecular entity on the plate and then should also bind very cleanly to that in an animal model, it's most likely way more complex than that, right? We got a certain cellular surface state that must be present for that to happen. So glycosylation patterns, membrane composition, complexing that must reveal the epitope and not be shielded, and so on. So if you can conclude anything so far, basically on this data, is that context matters, right? It is really important that we think of whether or not our epitope is actually accessible once we challenge our peptide in vivo. And our idea to overcome this is to do more in vivo phage display, making sure that we set up biopanning strategies to select for peptides, build that into that biopanning strategy, build that in so we can get peptides that behave well in vivo as well. Okay, and with that, of course, I also want to thank, first of all, Jessica Newton-Northup. She is my business partner, co-founder of Cell Origins, and she's done a lot of this work, and so has Dr. Malika Assar. At the University of Missouri, we've collaborated a lot throughout the years, and my early work that I presented was done in my PhD advisor's lab, Dr. Susan Deutscher. Of course, we also want to thank Dr. George Smith, first of all, because he invented phage display, which has been great for us. Very interesting research throughout like over 40 years now, but also because we continue to use his libraries, some of which we've modified for these studies as well. Dr. Austin Prater, who has done the bioinformatic analysis for us, and then the NIH for providing SBIR funding for this study. So thank you very much.
Thank you so much, Matt. So if you have any questions, feel free to drop them in the chat or in the Q&A. And then I will let Stan go ahead with his questions.
Okay.
Yeah, well, thank you very much, Mette. That's super comprehensive and super, super interesting. I have lots of questions touching very different aspects here, but let me start with the first one. So since I'm working more about the data, my first question that I have when you mentioned that you're doing like typically 3-4 rounds of painting, yeah, with the phage. So my question is, that's actually what I see, like people trying, because it's expensive. 4 rounds, especially since you're doing in vivo with mice, yeah, it's like expensive. Do you think that it is possible to do less rounds but compensate this on the data analysis side? I don't know, with AI or some tools.
I definitely think that's possible. I don't even think you necessarily need AI for it. So when I showed the in vivo mouse study we did, that was done, it was published in 2014, right? So it was done quite a bit ago, and we were analyzing the phage individually with Sanger sequencing back then. So back then, the idea was we need to go out to 4 rounds to be able to actually see the differences and have peptides come out, right? So to stand out to us. I don't think we need to do that anymore, the 4 rounds. It really depends on what the target is and how we're setting up the phase display. But doing 4 rounds of identical selections with the NGS, like the depth of sequencing we have
Mm-hmm.
nowadays, and for example, the type of software that you provide, it's not necessary, right? So we
Mm-hmm.
can save both time and money there.
Mm-hmm. And in general, are there like some substantial differences with this in vivo painting and in vitro when you have like a plastic? I mean, the wet lab on the data side, is it like, I'm curious from the software side.
If
Do we
we
need
if
to
we get
adjust our,
if we
yeah.
get plastic binders, you mean? Is that what you're asking?
Yeah. So not the mice when you do this in vitro, not in vivo.
Yeah, so you always, I mean, you need to run the proper controls. So we
Mm-hmm.
always do a negative selection basically against the plastics, or we're using beads or anything like that to immobilize the
Mm-hmm.
protein. You got to do that because you're going to get a lot of background binders, and by that I mean specifically plastic, right? Any hydrophobic amino acids love that. The beads as well. If you're using streptavidin, BSA, you basically got to go in and basically run a proper control to make sure you don't get those. And sometimes it's necessary to do it more than once if you have a very challenging target.
Mm-hmm. Mm-hmm. Mm-hmm.
Yeah.
Thank you. And talking about the targets, what really super interesting for me is your like blind method without like knowing the target in advance, but then finding
Yeah.
the target. Yeah, and here in this blind discovery is I think it can be really comprehensive and much better than like you can find something which is much better than you assume. But then the question is, do you frequently or in general, whether it's frequent situation when you find something completely unexpected when you do this blind discovery?
I don't know if I find anything unexpected. Um,
Mm-hmm.
could you do like what do you mean by that? Like a binder, a strong
Mm-hmm.
binder, or
Yeah.
some
So
like a
when you're,
targeting? Yeah.
yeah,
So one
I mean
thing I think,
the
sorry
target.
to
I mean the
interrupt. Yeah.
target.
Okay.
I mean
Yeah.
the.
So one thing most people think of targets as proteins, but it's rarely lipids because it's really difficult to do
Mm-hmm.
lipid selections, even though it can be done. I know Pascalini has done that, but carbohydrates. So
Mm-hmm.
uh, thinking of carbohydrates, especially in relation to cancer, right? That the glycosylation patterns are so different. I know that Jessica here in Northopsy has worked with a peptide that targets a TF antigen, which is a
Mm-hmm.
disaccharide. So
Mm-hmm.
a lot of the time we will get peptides that bind to like the carbohydrates, the oligosaccharides on the proteins.
Mm-hmm. Mm-hmm.
Yeah. So I don't know
Mm-hmm.
if that surprises me, but I think that often we think of the protein as being what we're targeting, and in reality, could be the carbohydrates.
Mm-hmm. And when you're identifying the target, so that you describe the CRISPR knockout method, yeah?
We
To,
haven't found that yet. That's what we want to do. So we're still analyzing the data because we have enormous amounts of data since we also have multiple peptides.
mm-hmm.
So that's what we're currently doing. Of course, eventually we want to find out what the actual target is.
Yeah.
So I think one of the next steps is definitely to do the classic pull down assay and mass spec. And I'm going to expect that we're going to pull out a complex, right?
Mm-hmm.
So a
Mm-hmm.
complex of proteins. That would be
Mm-hmm.
my expectation if it's going to correlate with the data that we
Mm-hmm.
already have. Yeah.
Mm-hmm. 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 Okay. Interesting. In general, when you don't know the target, like I mean, as I understand the world of CROs, when you have a customer coming to you, the customer already
Mm-hmm.
has the target, yeah, and
Yeah.
knows the
Well,
target.
most of our clients have targets. That's the way it typically works out in biotech industry. People are developing peptide drugs, and I'm not against people choosing a target. I think that's great if you have a target. I just think
Mm-hmm.
we need to think about that target in relation to what it's actually like on the cell surface.
Mm-hmm. Mm-hmm. Mm-hmm.
Yeah, it
I
makes it
see.
easier for us, right? If we do know the target in the way that
Yeah.
yeah, we can kind of customize the selection strategy and the elution strategy a little bit better. We don't have to go in and determine what the target is later.
Mm-hmm. Mm-hmm.
You can
But
do
then.
both, right? It's really like pick your poison almost. You're going to
Mm-hmm.
get your hurdle at one point. Yeah.
Mm-hmm. Mm-hmm. Okay. Yeah. Well, that's super interesting,
Thank
and
you.
Yeah, it's just few more questions. I see in general, like from our users who use our software, we have more and more requests for the peptide discovery. Yeah, so our
Yeah,
traditional area of interest is antibody. Yeah, and we
yeah.
have like pipeline for antibodies, but we're seeing more and more people are doing peptides. So is it a general trend in the industry, peptide discovery? And if yes, then why?
Yeah, so definitely, I believe that peptides. Last time I checked, peptides are the fastest growing, basically, molecule out there to be used as drugs. And that started with the GLP-1 peptides, right, for obesity and
Hmm.
diabetes. And back when we started out, so that's 15, 16, 17 years, Jessica, a little bit longer. A lot of people didn't believe in peptides. I've had reviewers tell me that peptides can't bind to proteins, which is a little
Hmm.
bit weird because there's a lot of hormones
Yeah.
out there that are peptides and work naturally, right? But it was the GLP-1s that I think really got scientists to realize, and mostly investors to realize, okay, we can actually use this, and now we can use peptides to target cancer. They have certain advantages over antibodies.
Hmm. Hmm.
Antibodies also do that, vice versa. We need to use them for most likely different applications, but they're just as
Hmm.
important and can be developed to be just as specific.
Hmm. What are the advantages over the antibodies?
Sorry, could you repeat that?
What are the advantages of peptides over the antibodies?
So, especially when it comes to radiotherapy, radioligand therapy, like you can see,
Hmm.
was my study. Here we labeled with a radioligand.
Hmm.
It's very important that we get no off-target binding. It's very important that the bias distribution is very fast. We want the radioligand to get to the tumor immediately to deliver the payload of the radioisotope and then get excreted very fast. So peptides have very fast biodistribution compared to
Hmm.
antibodies. Antibodies stay in the body for days, and that would lead to very high toxicity if there's a radioisotope on there. So
Hmm.
specifically for radioligand therapy, which is also a very fast growing field right now. It's very exciting. It's exciting actually to be a part of. So their antibodies are really not great for that, and for most of
Hmm.
the therapies, and that's because of that extended biodistribution. They just stay in the body for longer.
Hmm. You also mentioned the off-target binding. So for peptides, this is better. I mean,
No, not necessarily. So I will say generally we can get antibodies to have higher binding affinity and specificity, right? So we can still get off-target effects with peptides, but there's ways around that. For example, we're doing studies like this. The
hmm.
thing is that peptides, they can extravasate into the tumor a lot better as well. So we kind of overall, the advantages lay in the pharmacokinetics of peptides, especially when it comes to radioligand therapy.
Hmm. Hmm.
Yeah.
Hmm.
A challenge,
Uh,
for example, is kidney uptake then, because you can see peptides excrete through the kidneys. So the drug's got to get out of the body somehow, right? And the challenge here is that sometimes peptides bind to the kidney, and at the very least, they get excreted there, and that leads to toxicity.
hmm. Hmm. And so one other last question actually I have is again since my background is primarily from antibodies, yeah. So in antibodies, when we find the good binders, yeah, even I mean good binders in vivo. The problem which stays is the developability of those antibodies and ability to
Yeah.
produce them downstream. Do you have the same
Yeah.
problem in the peptide world?
Generally, no. So generally,
Hmm.
peptides are not, listed an immune response, although it
Hmm.
can happen. So if the immune response starts, it's typically a lot smaller than with antibodies. So
Hmm.
being injected multiple times tend to not be a problem in the same way.
Hmm. Okay.
Yeah. Yeah.
Thank you.
So that's
Thank you
another
very much.
advantage. Yeah. Yeah.
Okay,
Another thing I
awesome.
can think of is, sorry, I just wanted to mention that peptides are now also being developed to go into the cell and target cytoplasmic proteins.
Hmm.
So that's something that can be difficult for antibodies to do if they get internalized, they get into endosomes.
Hmm.
Mhm.
Yeah.
Okay, great. So I actually just wanted to ask you one question myself, Matt.
Yeah,
So I think it's really interesting because you have this experience in academia. You did this for your PhD project, and then also currently as a professor. And on the other side, you're also working on this in industry where phage display is a lot more popular. So can you talk
yeah.
about the differences in methods and maybe the differences in the end result? So if for academia, maybe it's more exploratory. For industry, you're trying to get a drug to market. Can you talk about the differences in the methods, applications, the maybe differences in also timelines as well?
Yeah, I think overall the strategies are the same, right? So a biomarker is chosen and a phage display selection strategy is set up. The differences comes to funding and time. Like
Mm-hmm.
that's generally industry has more money. They can put more money into more advanced biopanning strategies like in vivo work. Whereas there's of course less funding in academia for things like that. But overall, there's a lot of great phage display being done in academia as well. I don't want to say that at all that doesn't happen. I think the major hurdle, and this happened to myself when I was in academia and graduate school, is that you want to publish and you can be very quick to publish peptide sequences. And once you've done that, you lose IP, right? So if you have the goal of taking that further, maybe into developing a drug or an imaging agent, then you need to talk to university and protect your peptide sequences. And not all universities are, easy to talk with, although I think they're getting better and better at it. So I think that's the main difference. Make sure you get IP. If you're doing phage display, get IP on your sequence if you can, because
Mm-hmm.
else you can't use it for anything. That's the thing with J18 here. I think it's actually a great peptide. I would love to have taken it further,
Mm-hmm.
but we published and we needed to do that. There's nothing wrong with publishing, but we should have protected it first.
Mm-hmm.
Great, that's good advice. And then actually just one more follow-up. How did you decide to open up Cell Origins and how did you decide to take that work into industry?
Yeah, so Jessica and we have been friends for many years. We were like in the same lab when I was in graduate school. And we basically wanted to create our dream job. So we were very passionate about a phage display and very
Mm-hmm.
passionate about peptides and have been for decades. So we basically wanted to come together and say, let's find an SBIR we can apply for. And we were awarded that. And that's how Cell Origins got started. As I think a lot of people have experiences with SBIRs, is that the money runs out pretty quickly from that phase one, even before you can get to submit to phase two. There's that kind of a dip in funding. And that's when we started to think about maybe we could make money also offering our services and our experiences. There's few people that have more experience in this field than we do. So we actually that's what we did, and we had people contact us. So that's how we make our money now. We still have our own R&D developing peptides, but we pay the bills, so to speak, doing partnerships, collaboration, CRO work with other companies, and also academia. So if anyone is out there in academia, there are some libraries that are almost like hardly any charge to purchase. And if you want advice, anything, everyone feel free to reach out to me, right? I always love to talk science. So if you think it's too expensive for you to do phage display, please reach out to me and we can talk about that. I think it's more accessible than you think it is.
Great. All right. So I want to wrap up today's conversation. Thank you so much, Matt, for joining our High Affinity Talks. So we'll send out this recording to all of the attendees afterwards. And we're really glad that you were able to join us today.
Well,
Yeah,
thank
it's
you so
super
much for
insightful.
inviting me.
Thank you
Awesome.
very much.
Thank you.
Thank you.
Bye.
Bye.